Hi everyone, welcome to medical microbiology. So today we will discuss chapter 14 and 15. So these two chapters talk about human defense mechanisms. Therefore, I combine these two chapters together in one lecture. Okay, first of all, we have to understand our natural defense mechanisms, which is consists of white blood cell. Okay, so this is what we call the armies, just like the armies in our country defense mechanisms. So this is the people, the armies that uh, fight against infections. There are five types of armies for white blood cells in our system. The first one is called the neutrophils. These neutrophils are the white blood cells that usually fight against bacteria. The second one is called the lymphocytes, and this second type of armies, they usually fight against viruses. And then the third type of white blood cell is called the monocyte, or sometimes it is called also macrophages because they are very big white blood cell, and they usually also fight against bacteria. The next one is eosinophils. The eosinophils, they usually fight against parasites like worms and protozoa. And then the last one is called the basophils, and they usually fight against allergens, and they produce what we call the allergic reaction. So these are the five type of white blood cells. Okay. I put this uh, list is in sequence, which is from the most abundant ones to the least abundant one. So neutrophil is actually the most abundant type of white blood cell in our system. The lymphocytes going to be the second abundance type of white blood cell, and then monocyte, eosinophil, and basophil. So basophil is the least abundant type of white blood cells in our system. In addition to the army, in our body, we also produce the weapon. So remember, the weapon is actually produced by this army. So there are some army that produce special weapons. Okay. The main weapons is called the antibody. Okay, so the antibodies is actually the main weapons in our body. And these antibodies, they are produced by the lymphocyte, especially the B cell. So lymphocyte consists of two types of cell, B cell and T cell. So these B cells are the one that produce this antibody. Okay, there are five types of antibodies in our system. Okay? Sometimes the antibodies is also known as immunoglobulin or Ig. That's another name for antibody, Ig or immunoglobulin. So there are five types of antibodies. The first one is called the IgG. That's the most abundant one. The second one is called the IgA. The next one is called the IgM. And the next one is called the IgD. And the least abundant one is called the IgE. Okay, so again, these weapons or the antibodies is produced by the B cell. These antibodies is also fight against some specific infection. For example, IgG mostly fight against any type of infections, okay, whether it is coming from bacteria, viruses, parasites, fungi, and etc. Okay. The IgA, this is also fight against infection, all type of infections. However, this IgA usually located in the fluid, not really in the blood. So this IgA located in the fluid that we produce, like for example, in the tears, mucus, sweat, there are IgA in this fluid. Okay, so IgG located in the blood, while the IgA mostly located in the secreted fluid. The IgM, okay, this is the next abundant ones in the blood. They usually fight against foreign cells. For example, if we have blood transfusion, so we receive the cells that coming from other people, and this cell is considered as a foreign cell, and usually the IgM will try to fight against this type of antigen or foreign cell. The IgD, they usually have other antibodies or either other immunoglobulins. And the last one, the IgE, the least abundant one, this is the one that usually also help to fight against allergen. Okay, it's kind of work together with the basophil. You see the basophil and IgE, they usually fight against allergens, the antigen that cause allergy. Okay. There is additional weapon that also produce in our system. Okay. The first one is called the interferons. This interferon is usually produced by the infected cell. So if we have the cell that is infected by viruses, for example, this infected cell able to produce interferons. And these interferons then will go to the healthy cell, try to give warning to the healthy cells about the virus or about the pathogenic microbes that enter into our system. The next one is a proteins. A group of protein is called the complement system, and this usually fights against bacteria. So help us to fight against bacteria. And the last one is toxin. This toxin will also fight mostly against viruses. Okay, so remember the antibodies, the five type antibodies is produced by the B cell. The toxin mostly produced by the T cell. So this T cell produce the toxin that will destroy the viruses. The way they destroy the virus actually by destroying the infected cell. So this toxin also uh, produced by the T cell, it also will destroy the cancer cell. So the cancer cell usually fought by the T cell. It's destroyed by the T cell by producing the toxin. Okay, again, we review the armies, which is the white blood cell. Again, there are five types of white blood cell. The most abundant one is called the neutrophil. About 55 to 70% in our blood. Okay, so it is a neutrophil. These neutrophils is used to fight against bacteria. So when the neutrophil increase in number, for example, you take the sample, the blood sample, and calculate the neutrophil, lymphocyte, monocyte, eosinophil, basophils. So the natural ratio is between 55 to 70%. However, if from your sample, for example, this neutrophil increase into 90%, then it will give us the indication that there is a bacterial infections in our system. Okay, so it can be used for diagnose infection. Okay, the second type of white blood cell is called the lymphocyte. This is the second most abundant type of white blood cell. And again, the lymphocyte consists of B cell and T cell. They usually present in about 20 to 35%. So this is for healthy blood, 20 to 35%. Now, for example, if we take a sample and then lymphocyte is actually ratio actually increase, let's say into 50%, then it is indication that there is a viral infection. Because what? Because this lymphocyte fight against viruses. So increase the number of lymphocytes give us indication that there is a viral infection. The next one is called the monocyte or macrophages. This is the one that also fight against bacteria. And the normal ratio is between 3 to 7%. Again, if we take a sample, blood sample, and calculate them, let's say increase to 20%, this is indication that there is a prolonged bacterial infection. Okay, and the next one is called the eosinophil. The normal concentration is around 1 to 3%. And these eosinophil mostly fight against parasites like worms or protozoa. Now, if we take a sample again and then we calculate and this eosinophil increase, let's say, into 10%, then it gives us the indication that there is a parasite in our body. Okay, so this is how we use this differential calculation of white blood cell to diagnose disease. The last one is called the basophil, and this is very, very low concentration, less than 1% in our blood, and they usually use for fight against allergen, okay? The antigen that causing allergic reaction. If, for example, in our sample we calculate and this basophil increase to 10%, then it is an indication that there is allergy or allergic reaction. So people that have allergy, they usually have increased the basophil concentrations in their blood during the allergy season. Okay, so these are the main army that fight against infection or disease that present in our body. The study of body or human defense mechanism is called the immunology, especially when we study the lymphocyte over here, okay, which is the B cell and the T cell action. Okay, so this is called the immunology, the special scientific study of immune system. So for the healthy humans functions, the immune system usually have three main action. First, they usually survey. They always survey our body. So this is what we call the surveillance. So like a T cell, they usually go around our body using the blood circulation and checking if there is something happen in our body. Like for example, if there is cancer cell developed, then the T cell will destroy that cancer cell as soon as possible. So this is what we call the surveillance. So that's the first action of healthy immune system. And then when they survey, then they might recognize if there is a foreign material. This foreign material is also known as antigens. So they will recognize it. And then these antigens or this foreign material then will be destroyed by our immune system. So this is what happened in our body. It's always happening, which is the immune system will survey our body, our system. If there is foreigners, if there is antigens, then they will destroy that antigen. They will recognize it and then they will destroy it. And when the antigen destroy, then we become, we keep our body healthy. But if we are unable to destroy that antigen, then we have the disease. So sometimes we have to take drugs from outside, like antibiotics, in order to help our immune system to fight against these stubborn antigens, like maybe bacteria or viruses. So antigens can be anything that actually causing disease in our body, provoke the immune system in our body. That's what we call the antigens. It might be bacteria, viruses, allergens, worms, parasites, or even sometimes our own cell become antigens in our body and causing the disease. So the disease that caused by our own cell, our own antigens that coming from our own body, it is called the autoimmune disease. So the disease that caused by our own cell. Okay, again, this is the process. So the immune system, especially the T cell, will always perform the surveillance. Okay, surveillance. So, okay, if we talk about our community, maybe this T cell, they work as like a sheriff. Okay, like a sheriff or police that always survey the neighborhood, check if there is something happening. So this sheriff or this white blood cell will check uh, if something happened or not. If there is nothing happen, so they check this normal cell, and then nothing happened, and say, okay, there will be no reaction. You okay? You fine? Okay? And then this white blood cell then will survey other area. So just always like that. Do the surveillance. If, for example, okay, this white blood cell see that this one is not our own cell. So this not our own cell will give special markers over here. And then this white blood cell, okay, for example, the T cell, we recognize this foreigner. Oh, this is foreigner. And then if it is foreigner, then this white blood cell will destroy this foreigner. So destruction. Okay, so this is what happenings in our body over and over. Surveillance, then recognition. If it is just our own marker, then there will be no reaction because this white blood cell will not kill our own cell. This white blood cell will not destroy our own cell. But if this one is foreigner, so it will be recognized with the white blood cell. Why it is able to recognize? Because the marker will be different over here. Okay? So the markers of the foreigner cell will be different than the marker of our own cell. So this one, the cell will be able to recognize this foreigner, and finally they will destroy this foreigner. Okay, and if again, if we're unable to destroy these foreigners, then we have the disease, and sometimes we will have to take medicines or drugs from outside in order to destroy this foreigner or this disease, the pathogens. Okay, okay. Based on that, then we can actually classify our host defense mechanism. So our defense mechanism, this is human defense mechanism, can be divided into two main types. The first one is called the non-specific or innate. So what is non-specific mean? It means that these defense mechanisms will fight any type of infection. It is not specific. So any type of infection, any type of infection. Okay, so it fights against any type of infection. This is what we call the non-specific. Innate, so innate mean, okay, we have it since we were born. So we have this type of immune or defense mechanism with us since we were born. So we born with this non-specific defense mechanism. Okay. The second one is called specific or acquired. Okay. So specific mean is only fight against specific infection. Okay. For example, there is a specific defense mechanism that only fights against chickenpox. It cannot be used to fight against polio, as it's only one type of infection. Fight against one type of infection. Okay, so one type of defense mechanism fight one type of specific infection. So we have several type of this specific uh, defense mechanism. And these ones acquire. It means that it has to be made. Okay, it has to be made. It is made when we have infection or the disease. Okay, so compare with this innate over here. So we have it since we are born. We born with this innate defense mechanism. So we have it whether there is infection or not. We have this type of defense mechanism, which is non-specific and innate. For the specific ones, we only made this type of defense mechanism whenever we have the infection. For example, we have a chickenpox infection, then we will make the defense mechanism that will fight only against chickenpox infection. Okay, now the non-specific defense mechanism can be divided into first line of defense and the second line of defense. The first line is mostly used for prevention. Okay, so prevention or for blocking the entrance of disease, right, or infection. Okay, so this is for blocking or prevention. The second line of infection, yeah, this is happening when the first line of defense mechanism fail, then the disease or the infections or the pathogen will enter into our body. Then the second lines of defense mechanism will try to remove it. Okay, so this is the action, okay, after the infection enter to our sterile tissues. So we have what we call the second line of defense. If the second line of defense fail, then we still have one more line of defense. It's called the third line of defense. And this third line of defense is actually part of the specific defense mechanism. Okay, so this is the process, of defense mechanism. Start from the blocking the entrance, which is the first line of defense. Then if it is fail, then it's go to the second lines of defense. If the second line fail, then it will go to the third lines of defense mechanisms. Okay. Now, what are the first line of defense mechanism? Again, this is a prevention, the blocking of the entrance of microorganisms. It can be done physically, okay, by the physical barrier. Like for example, our skin. Okay, our skin is very tough. It has many layers of cell. So it's become physical barrier of infection. Okay. Another example is in our nose, for example. There are hair, there are mucus that actually become physical barrier for entrance of many microorganisms. Okay, so the microorganism will be filtered by this hair and also the mucus in our nose. So this is example of physical barrier of the first line of defense. It can be also performed by the chemicals that we produce. Okay, like for examples, in our sweat, there are enzyme, which is chemical, it's called the lysozyme. And this enzyme is actually able to destroy the microorganism. The sweat itself is physical barrier because when we are sweating, we actually physically remove some bacteria or microorganism. Spit, for example, also physical barrier. Coughing, sneezing, also physical barrier. Tears that we produce, this is also physical barriers at the first line of defense mechanism. But in the tear itself, there is a chemicals. In the sweat, there is a chemical that able to destroy microorganisms. That's become the chemical barrier. Okay, and then the last one for the first line of defense is genetic barrier, which is some people they have special gene that help them to protect against some disease. For example, people that live in malarian belts in Africa, they have special gene that actually protect them against malaria. So if this gene present, then they will be protected. If it is not, then it will not be protected. So this is kind of genetic barrier for some people that have a special genes to fight against specific infection. So this the first line of defense, which is mostly prevention or blocking the entrance of disease. If again the first line of defense fail, then the disease, the infection will enter into our body. Then we have the second line of defense. It starts with the inflammatory responses. Then there will be interferon production, phagocytosis, and also complement reaction. This is part of the second line of defense. We will talk about this one later. Okay. And if the second lines of defense fail, then we still have one more, the third line of defense. And this is type of specific defense mechanism. It is done by the T cell and also by the B cell. Okay. Then we will talk about this third line of defense later. So this is a summary of human defense mechanism, which is divided into two types. The first one is called the non-specific defense mechanism. This type of defense mechanism will fight against any type of infection, and we born with it. Therefore, it's called the innate. And this non-specific defense mechanism consists of two lines, the first line and the second line. And then the second type of defense mechanism is called the specific defense mechanism. It's only fight against specific infection, and it is acquired. It will be made after the infection or during the infection. Okay. So this specific defense mechanism is also become our third line of defense mechanism. Okay, so we start with the non-specific defense mechanism, which is this is actually discussed in chapter 14. So non-specific host defense, and this is also called the innate defense mechanism. Okay, we actually born with this defense mechanism. Okay, so the non-specific defense mechanism is also known as innate defense mechanism. Okay, again, it consists of three types. Okay? The first one is called the first line of defense. It consists of physical barrier. And again, we actually have a lot of examples of physical barrier or the first line of the first lines of defense. So this is actually the action that will protect us against the entrance of the disease. Like for example, the skin structure. It has so many layers. So our skin, if it is healthy skin, it will protect us against the entrance of many pathogenic organisms, microorganisms. But whenever there is a break in the skin, okay, maybe there is a cut in the skin, then it is actually the place where the microorganism will enter into our steroids. So make sure when you have a skin cut on the skin or damage on the skin, make sure you protect this, you take care of this damaged skin. Otherwise, it's become the place for the entrance of microorganism into our sterile body, especially to children, because sometimes children just don't care. So as a parent, then you have to take care of this problem. You put something that will protect the skin from infection. Sweat. Okay, when we sweating, yeah, this is also physical barrier. We actually remove a lot of microorganisms from our body. Mucus, like for example, mucus in our nose, it will bind many microorganisms. It kind of protects them from entering into our sterile tissue. When we blinking our eyes, then produce the tears. That's actually also remove a lot of microorganisms from our eyes. Uh, that's the physical barrier. Uh, I think this is not physical. Stomach acid is not physical because acid is chemical. So we will put that on the next one. The hair, okay, hair in our nose, for example. This is also uh, filtering mechanism, which is physical barrier of our mechanism. Another example is sneezing, okay, coughing, urination, defecations. That's all example of physical barrier of the first line of defense. Because when we sneeze, when we coughing, when we urinate and defecate, is actually remove a lot of microorganisms from our body. So it's kind of protect us, okay, from maybe infection. The second first line of defense is called the chemical barriers. It is done by chemical. Like for example, oil. Okay, so oils in our skin, they actually able to kill many type of bacteria. Lysozyme, this is enzymes that we produce and present in the tear, in the sweat. This lysozyme will kill a lot of bacteria. The tear and the sweat itself is part of the physical barriers, but the enzymes that is present in the tears and the sweat is part of the chemical barrier. So it's actually double uh, mechanism, okay, from physical and chemicals at the same time. Uh, High lactic acid and electrolyte concentration in the sweat, in the tear, they're also able to kill bacteria. Skin sometimes it has acidic pH, uh, hydrochloric acid in the stomach. So this is also chemical. There are so many bacteria that will enter into our stomach through the food and the drinks. And because stomach is very, very acidic, it actually kill a lot of bacteria or viruses that coming from outside into our GI tract. Okay, so this is very important over here. Okay, the hydrochloric acid in the stomach. The digestive juices, enzymes, and bile in our intestine is also able to destroy some type of microorganism. Cements also contains some antimicrobial chemical that will destroy some uh, microorganisms. The vagina also have the acidic pH, which is also help that structure to kill several type of microorganisms. So this is chemical barrier. Okay, so I think should be one more the genetic barrier. So this, the chemical barrier, I believe in the textbook is mentioned about genetic barrier. Okay, so example, some people that live in the malarian belts in Africa, they have special gene that able to protect them against malaria infection. Okay, this is the picture of some first line of defense, like for example, the skin structure. Okay, the skin structure, the sweat glands. Okay, production of sweat, and lysozyme at the same time, wax in our ear, and also protection against some entrance of bacteria. Low pH in our skin, cilia. This is a physical barrier in our respiratory tract. So the cilia actually remove many bacteria from lower respiratory tract into our mouth. Saliva is also have lysozymes on it. Mucus in the nose, tears, and then oil glands in our skin. Urination, defecation, that's also part of the first line of defense mechanism. Stomach acidity, this is also part of the first line of defense mechanism. So we actually have this type of defense mechanism even before the infection. So this is the protection, this is the blockage against the entrance of microorganism that might cause disease to us. Yeah, this is the third one over here, the genetic barrier. C. Okay, so again, some people have the ability to protect themselves against disease based on their genetics differences that maybe exist in some community. Okay. So examples is people that live in the Malarian belt country in Africa, they have special gene, it's called the sickle cell gene. Okay, so let's say normal gene is like this. Let's say normal people carry this type of gene, normal. So they usually normal, okay. And then people with the sickle cell gene maybe carry this sickle cell. So they, this person actually will have what we call the sickle cell disease, which is a type of anemia. Okay, and but if they combine, let's say like this, and this one is heterozygote, this person usually able to is protected against malaria. Okay? So people that have normal gene usually is not protected against malaria. It's not protected against malaria. So they will usually maybe die because of the malaria. So most people in Malarian belt country, they carry this type of gene, which is the heterozygote type. So when they move to United States, for example, okay, some of them maybe, let's say this one married with another one that carry heterozygous gene, then they might have children that carry both of the sickle cell gene. So when these children have both sickle cell gene, then these children will have disease called the sickle cell anemia. Okay, so mostly the sickle cell anemia present in the African American people because they are, their parents or the forefathers that come from African country carry this sickle cell gene with them. Okay, so we're done with the first line of defense. Now we go to the second line of defense. Okay, so the second line of defense, it consists of several actions. Start with the, of course, recognition. Okay, so recognition, so when the antigens, when the pathogenic disease or infection coming into our system, they will carry special markers, and then our immune system will be able to recognize it. And then after that, yeah, start the first process is called the inflammation. Okay, so whenever we have, for example, damage or infection, that site of infection usually become inflamed because this is actually the first response of the second line of defense, which is the inflammatory responses or inflammation. Sometimes it's also accompanied with fever, okay, higher body temperature. The next process will be phagocytosis, okay, which is uh, distractions of pathogens by phagocytes or cell eaters, the cell that able to eat pathogen. It's called the phagocyte, and the process is called a phagocytosis. And also our cell will produce the interferons. Usually the infected cell will produce interferon that will give warning to the healthy cell. And also our cell will produce what we call the complement system that will destroy the pathogen. And we'll see one by one the process. Again, the recognition, it is done by mostly the T cell. Okay, let's say this is a T cell. So this T cell actually have special proteins that able to recognize the foreigner cell. Okay, let's say this like this. So this is the proteins that able to perform recognition. Let's say this is foreigner cell, or like maybe uh, pathogens, or some mostly it's called the antigens. Okay, so anything that provoke the infection or immune system. This one will carry special sign or marker. Okay, that will be recognized by this receptor cell. So this one receptor. Okay, so the T cell have receptor that will recognize the marker. So when it is recognized, then we know there is a problem. Okay, so this is the first step, which is recognition. And this then go to the second step. Okay, second action, which is the inflammatory responses. So whenever we have infection, we will have this response. It's called the inflammatory responses or inflammation. There are several signs and symptoms of the inflammatory response. Of course, redness to the area of infection, they usually become more red. This is what we call the redness. Why? Because this area that have infection usually will receive more blood, because blood is the one that carry our immune system, the one that carry white blood cells, eh? Neutrophils, lymphocyte, monocyte, they all carry by the blood. So therefore, when the blood come into this inflammation area, that place become red. So this is what we call the redness. And that area also become warmer, okay, warms, because the action that happening in that area where the white blood cell try to destroy the pathogens, the antigens. And then it will be swelling, okay, inflamed. This is the area become bigger. So this is also the sign of inflammation. Okay, warm, redness, swelling, pain. Okay, this is the symptom, which is some when we have an infection, sometime in that place we feel the pain or maybe itch, and there is a possible loss of function on that side. So that those are the sign and symptom of inflammation, which is the second response of second lines of defense. Again, this is just examples when our tissue get injured, boom, the pathogens, bacteria will be enter over here, and then our blood cell will carry a lot of blood, white blood cell into the area, then it's become redness, okay, become red, and then it's also inflamed. Okay, so this is white blood cell over here. They fight this bacteria. Okay, so when they're able to fix the problem, then what happens? We mostly have what we call the scar. So scar formation over here after these white blood cell able to fight against this infection. Okay, so this is several action that happenings when we get the infection in specific site of our body. Now the leukocyte, which is the white blood cell, so this is another name of white blood cell is leukocyte. So some white blood cells have a unique characteristic. Okay? For example, they're able to move from blood circulation into the damaged tissue. So remember, they are carried by the blood, but they're able to move out from the blood vessel. This is what we call the diapedesis. So the action is called the diapedesis. And they also have the ability to move to another side based on chemical sign. Okay? So chemotaxis is mean that the movement due to the chemical sign. So the white blood cell able to migrate or to move to certain area when they sense the chemicals on that area that maybe is not present during normal time. So this is what we call the chemotaxis. So they're able to move based on the chemical clue, and they're also able to move out from the blood vessel into the infection site. Okay, so this is examples of diapedesis. So the white blood cell carried by this blood vessel. Okay, they able to actually move from this white blood vessel out. Okay, so move out from the water. They're able to squeeze between this epithelial tissue of the uh, blood vessels. And they usually move to the infection area where the infection area actually give the clue by producing the chemicals. So remember last time we talked about the bacteria or pathogens, they produce the virulent factors like enzymes and toxins. Okay, these are actually chemicals. So these chemicals give the clue for our white blood cell to come to this area and fight them, okay? Fight the infection. Okay, the next one is fever. So this is also the second part of the second line of defense. Usually when we have infection, then we also have fever. So this is caused by the pyrogens that may be produced by the pathogens, and this pathogen actually will set our thermometer in our brain. So there is a special area in the brain that set our body temperature. When the pathogens enter into the setting place, then it's increase the body temperature. So we have what we call the fever. So fever actually have benefits from us, okay, which is it will inhibit the multiplication of some bacteria that sensitive to temperature. Okay, so it increase also metabolic rate because increased temperature will increase the metabolism. Okay, so that's the benefit of fever. So if the fever is 101, for example, or maybe 100, that will be still a good fever. So this is benefit us. But if the fever increase to 104, 105, that will be dangerous. So we have to take the fever reducer when it's increased so high. But again, just remember, fever is actually part of our defense mechanism because they're able to kill some bacteria that's sensitive to high temperature, and also increase the metabolisms, the fight of white blood cell in the infection area. The next process of second line of defense is called the phagocytosis. That's the process called. It is performed by phagocyte. Cyte means cell, phago means food or eat. So phagocyte is the cell that eat bacteria. We have three types of phagocyte in our body, which is actually our white blood cell. The first one is called the neutrophil, which is the most abundant type of white blood cell. So they also able to eat bacteria. The second one, eosinophil, they able to perform the eat against the parasite. Okay, and then macrophages, this is the big eater. Okay, this is the biggest eaters. This is the one that actually able to eat so many bacteria in one time. So they kind of clean up the area of infection by eating the bacteria. So these are the three types of phagocyte in our body. And the process of eating of bacteria is called the phagocytosis. When you see the cis over here is actually mean process. Okay. Yeah, this is the process is called the phagocytosis. Okay. This is general activities of phagocytosis. First is survey the tissue that complements, they will recognize, there is a recognition over here. If the tissue of the foreigner cell coming, then they will know because they do the survey recognizing, and then they will ingest it, that foreigner, and then they will extract some information that coming from that foreigner. So immunogenic information that can be used later for us to even destroy more bacteria or pathogen in our body. Okay, let's say this one's here. So this is our phagocyte, which is white blood cell. Yeah. Phagocyte example is macrophages or the neutrophil. And this over here is bacteria. Okay, they are small. So this phagocyte, so you make it bigger over here, able to recognize this bacteria. Okay, and then it will be poo, making this mouth structure, and then eat them. When it is eaten, then it will be enter into the cytoplasm, and in the cytoplasm, this uh, vesicle, this phagocyte will produce the lysozyme, and this lysozyme will destroy this bacteria. And this one is bacteria being destroyed, and finally, gone is died. This can be removed from the cell. So this is how this bacteria able to eat and remove, the phagocytes able to eat, destroy, and remove the bacteria from our body, from our system. The next ones, okay, the next process in the second line of defense is called the interferon production. So this is actually a type of proteins that produced by the infected cell. So we're gonna see the process over here. Okay, let's say this is our cell that is being infected by viruses. So this is infected cell. This infected cell able to produce the interferon, and the interferon will be released into the healthy cell. So this is healthy cell, and whenever the healthy cell receive that information, the interferon, then it will make a protection against the infection that come into this cell. So it's preparing. Okay, just this infected cell, the interferon is just like a good neighbor. So when the good neighbor, for example, being robbed, then he will call the neighbor, his neighbor, and try to give the information, try to warn them, hey, I just get robbed. So make sure you lock, you close your door, the windows, and etc. So this is the interferon, just like a good neighbor that give information to the other neighbor when they get the infection. Okay, the last one for the second line of defense is called the complement system. It consists about 26 proteins that we produce, and these proteins actually will destroy the bacteria. Let's say this is bacteria. Eh? This 26 protein will attack this bacteria and then make a hole. Okay, on the cell wall. So when the bacteria cell walls destroy, eh, become the hole over here, then the water will enter into the cell. When the water enter into the cell, then the cell become bigger and finally will be exploded. Okay, because of this process. So this is the process of destruction of bacterial cell by the complement system. So this is the process. The proteins attached to bacterial cell walls, 26 or 20 of them, and then making the holes, okay? And then water will enter into the cell, and finally the bacterial cell become bigger, 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 and then boom, it's gonna be explode. Good, this bacteria will destroy. Okay, so that's chapter 14. In chapter 15, we actually talk about the third line of defense. It's called the specific immunity, which is become the third line of host human defense mechanism. Because this is specific immunity, it will be done by specific cell. So that specific cells are the lymphocyte. Lymphocyte, which is the B and T cell. Okay, so B cell and T cell. This is the reason why Another name of this specific immunity is called the lymphatic system or immune system. Because this is done by the lymphocyte, which is the B cell and T cell. Okay, B lymphocyte and T lymphocyte. So this B cell and T cell is not only protect us, is not only destroy the infection, special or specific infection, it's called the specificity, but it's also produce what we call the memory cell. So whenever we have the infection, special infection, specific infection like chickenpox, for example, then we fight them, and then after we fight them, we have the memory that will protect us against the second infection of chickenpox. So we become immune. Okay, we become immune to that specific disease. So this is the specialty of the third line of defense, which is the B and T cell, is not only fight against specific infection, but also produce the immunity. Okay, so we become immune against that specific infection. Okay, now the B cell, both of the B cell and the T cell, they are produced in the same organ in our body. It's called the red bone marrow. So red bone marrow is the place for B cell and T cells. The red bone marrow actually the place for production of all blood cells, including white blood cell, the red blood cell, and the platelets. And remember, white blood cell consists of 5 types: neutrophil, lymphocyte, monocyte, eosinophil, basophil. eh? And then lymphocytes, they have 2 types, which is B cell and T cell. So that's the reason why these cells are produced by the red bone marrow. When the B cell is produced, then it will stay in the red bone marrow to get mature. So it's become mature B cell in the red bone marrow. The T cell over here will move to the thymus gland to be mature. This is the mature T cell. Just for your information, both of them are produced in red bone marrow, but B cell will be mature in the red bone marrow. The T cell will be mature in the thymus gland. So the maturation place are different. The production place is the same. Okay, and then both of these mature B cell and T cell then will go to the special organ like the lymph nodes. This is the place where this mature B cell and T cell stay. So it's just like if they are army, so this lymph node is just like the place for army to stay. Okay, so B cells and T cell productions, again, both of them are produced in red bone marrow. The B cell maturation is present in the red bone marrow, so they stay there in the red bone marrow, while the T cell maturation takes place in the thymus gland. And this is the place for T cell maturation. Now, what is our enemy? So the enemies that enter into our system is called the antigens. So antigens are anything that enter into our body that might cause disease. Okay? It may be bacteria, okay? So bacteria, viruses, okay? Bacteria, viruses, maybe fungi. They are type of antigen. Or maybe for renal cell. Like for example, if we have blood transfusion, that the cell that enter into our system is become antigen, become our antigen. Or maybe part of the plant molecules, okay, like pollen, for example. That's allergen. Allergen is also considered as the antigen. Or maybe hair, dandruff, animal hair, part of skin of animals, or maybe some other things that enter into our body that have the potential to cause disease. They all call as the antigen. Okay, so antigen is anything that enter into our body that provoke our immune system. Okay, so there are some classification of the antigen. For example, alloantigens, and this is cell surface marker of one individual that are antigen to another that the same species. So, like for example, blood transfusion, we receive cell from other people. That type of antigen is called the alloantigen. It's coming from the same species. Super antigen is the antigen that will provoke our immune system, maybe coming from bacteria, viruses. Eh? Allergens, this is special antigens that provoke the basophil and the IgE, right? So remember, basophil is the one that fight against allergy, and the antibody that also help the basophil is called the IgE. So this is allergens. Autoantigens, this is our own cell that actually provoking our immune system. And usually, if it is happening, we have the disease, what we call the autoimmune disease. Like for example, RA, rheumatoid arthritis. This is a type of autoimmune disease when our antibodies is actually attack our own cell, especially in the joint area. Why? Because the own cell is actually produce the markers that our own antibody recognize this as a foreigner, even though it's actually our own cell. So that's what we call the autoimmune disease. This is just an example. There are some proteins, but when it is present by itself, it will not provoke the antibody. So this is not antigen. Eh? Remember, antigen is the one that provoke our immune system. However, if the small proteins bind to something, another protein, for example, then it's become bigger, then it will provoke the immune system. Then this one is become antigens. So antigens is anything that will provoke our immune system. Now, what's immune system that usually provoked by the antigens? They are called the antibodies. So antibodies will always try to kill or destroy antigens. Okay, so we'll always do that. So antibodies kill the or destroy the antigen. That's what we call the reaction. So sometimes we talk about the antibody antigens reaction, because that's the normal reaction of antibody, which is try to destroy the antigen. And just remember, we have 5 types of antibody. The most abundant one is IgG, IgA, IgM, IgD, and IgE. So every time we have the antigens enter into our body, then this into our body, then this antibody will be produced. So they are produced. Okay, they are produced whenever there is an antigen present. So these antibodies then will try to kill these antigens. So that's the process. So the antibody is not always present in our system. It is only made whenever we have antigens enter into our system. Again, this is the 5 class of antibody, the IgG. The monomer is only one type of structure, one side. The IgA, it is dimer, okay, 2 sides. The IgM is actually 1, 2, 3, 4, 5. So there are 5 sides of reaction over here. So therefore it's called a pentamer. So IgM pentamer. The IgD is also dimer, sorry, monomer, only one side. Okay, so this is one side over here. And the IgE also monomer. Okay. So IgG is the most abundant one, 80% in our blood. IgA about 20% or 30% in the liquid, okay, like mostly in saliva, sweat, tear, for example. IgM about maybe 6% in the blood, IgD about 1%, and IgE less than 1% in the blood. Right? This is the one that we talk about, that antibodies will always try to destroy the antigen. That's the process. So antibody is our weapons, the antigen is our enemy. So always the weapon will always try to kill the enemy. So this is what we call the antibody antigen reaction. So how? Now the question is how the antibody destroy the antigen. There are several ways. The first one is called the opsonization, which the antibody is actually coat the antigen, like the microorganism, and make them become inactive. It's not really destroy this antigen, but make the antigens inactive. It will not cause disease. Agglutination, this is when the antibody is actually bind to the whole antigen and then break the antigen and make them combine together, make a bigger molecules that will not be active. Get a large clumps of antibodies antigen reaction. And the last one is what we call the neutralization, where the antibody will bind to the antigen and then the antigen become inactive, become neutral. It will not be able to cause disease. So this is the some process of antigen antibody reaction. Okay. Again, this is the maybe the picture of that process. Like this one, for example, opsonization, when the antibody bind to the antigen and then make the antigens become available for the phagocyte. In this case, it's macrophages. So when these uh, antigens over here, which is bacteria, bind with this antibody, is become available for the macrophages to eat them. Okay, so this is what we call the opsonization. Neutralization, like for example, this antibody bind to the antigens like virus over here, and then causing the virus become neutral. You see the spike and everything will be covered by the antibody. Therefore, it will not be able to causing infection. This is the one that is normal virus over here, which is where it will be able to cause infection. But whenever this virus is covered by the antibody, it's become neutral. This is what we call the neutralization. Agglutination is when several antigens combine together with this antibody is become big clumps of molecule, and this big clump of molecule then will be precipitated, will not causing the infection. Okay, so it become inactive. Complement system, we still remember the complement system where the protein actually dig the holes in the bacterial cell. The same thing, the antibody is also able to produce similar action, which is digging the holes of the bacterial cell. This is the same name, it's become complement fixation. The other one is by combining these viruses with many antibodies. This is antibodies antigen reaction. So the virus is antigen, and then it's become precipitated. When it is precipitated, then it will not cause the infection or disease. So this is how the weapons, which is the antibodies, kill or destroy the antigens or microbes. Okay. The primary response, this is really important when we talk about COVID-19, maybe you've heard about this one, that the one that will increase, and the antibody that will increase will be the IgM. Okay? So the IgM will be increased first. That's the primary response. This is when somebody have a positive COVID-19 positive, for example, they're actually measuring the IgM antibody. After several days, then it's produced the secondary response. In this case, then the one that is produced a lot will be the IgG. Okay, so the IgG will be increased as the second response, and this IgG is actually the one that will destroy the antigens, like the COVID-19, for example. So when our IgG increase, that's a good sign. That's a good sign because that's the time when the antibody, in this case IgG, will destroy the uh, antigens. Okay, again, this is the first response. The IgM will be increased first. This is, then we have the positive indication, and then the IgG will be produced later. Yeah? And then in the second response, the IgG will keep steady produce. IgM actually will be reduced in production because they are not needed anymore. Mostly the IgG, the one that will fight these viruses. This IgG over here is the one that will fight against the viruses. So the first response usually take about 2 to 5 days, and the second response, let's say it is COVID-19, about 2 to 5 days, during incubation period. And then this one, the destruction of the viruses maybe take about 7 to 10 days. And this is when the IgG destroying the viruses. That's the time when we have the disease, the illness. Now for the specific immunity, again, this is the third line of defense. They are specially done by the special army, which is the B cell and the T cell. Okay? So there are several types of T cell actually. The first one is called the T helper, TH. Okay? Sometimes it's also known as a CD4. Okay? So they are developed from CD4 into T helper. The second one is called the cytotoxic T cell, or sometimes it's called TC. And they actually develop from CD8. And so when we heard about CD8, this is the same as the TC, cytotoxic T cell. Another type of T cell is called a natural killer. This is the T cell that lost their specificity, so they are actually able to fight any type of infection. But this T helper and TC are the one that is really important for our immune system for specific immunity. So the TC, when they destroy the infection, is actually by destroying the infected cell. So destroy the infected cell. So therefore, this type of immunity is called the CMI, cell-mediated immunity. So the TC actually destroy the cell. This is how they perform the action. Okay, so this is the TC, the cytotoxic T cell. When they have, let's say our cell have the infection over here, then the TC will produce the toxin and enzyme. eh? This toxin and enzyme then will enter into the infected cell and kill the infected cell. So this is what we call the CMI, cell-mediated immunity. So that TC is actually producing the toxin and enzyme that will destroy the infected cell, and the virus will be also destroyed. Some of them then will be available on the interstitial fluid, and this has become the uh, job of the antibodies to kill them later when it is outside the cell, because the antibodies is actually unable to enter into the cell. So antibodies like IgG, they only destroy the antigens that's located outside the cell. The one that is inside the cell will be destroyed by the TC. Okay. Now, this is just a summary examples of the how the immune system works. Examples, this is normal, during normal healthy cell condition. Okay, remember the TC will always survey. This is like a sheriff that always survey our body. If there is nothing happen, then say, okay, you okay. There is another cell that also perform surveillance. This is actually the T helper also performs surveillance. So I say we can call this as a deputy sheriff. Okay, so the T helper also survey, but T helper only survey the cell that able to eat bacteria, which is like macrophages, for example. The TC will survey other cell, all cell, except the macrophages and phagocyte and neutrophil. So all cell will be surveyed by TC, macrophages or phagocyte will be surveyed by T helper. So this is normal condition, there is nothing, so there will be okay. So it's just survey and then check, just like sheriff and deputy sheriff survey the community and there is nothing happen, then they say, okay, it's a good night. Yeah, have a good night and you say goodbye. Okay? But if there is something happen, like for example, chickenpox viruses enter into our system, then what happen? It will causing infection into this cell. So this is infected cell. So when the cell get infected, then it give the marker, okay, give the sign. So this is the time when the TC will know that, oh, okay, there is an infection, then the TC will become active. Yeah, when it is active, it will make other TC, it's called the TC clone, and also it will make the TC memory cell. Okay, now when the chickenpox enter into our system, the macrophages also know, okay, there is an infection. So these macrophages try to eat this infection, and it will be recognized by the T helper. Okay, remember T helper is the one that survey the macrophages. And whenever the T helper have that signal, then it will develop into T helper clone, which is active T helper, and T helper memory cell over here. Yeah. Right now we forget about the TC memory and T helper memory because it will work later. The one that work right now is the T helper, the active T helper over here. It will produce the chemical, especially it's called the interleukin. The interleukin will go to the TC, give the activation to the TC, and also give the activation to the B cell. So this is the B cell coming over here. When the B cell get the activation from the T helper, and then it will become plasma B cell and B cell memory. Okay. The plasma B cell, then the one that produces the antibody. Remember, antibody produced by the B cell, which is plasma B cell. These antibodies then will destroy the chickenpox viruses that is outside the cell. The one that enter into the cell then will be destroyed by the TC by producing the toxin and enzyme. It will destroy this infected cell, and then the virus will be available for the antibodies to be destroyed. Boom. This process happenings about 7 to 10 days. This is the time when people with the chickenpox infection will have the disease, about 7 to 10 days. The same thing with the flu, okay? Maybe 7 to 10 days. After that, the virus will be gone, and then we become better. We cure from the disease. Then the antibodies also will be destroyed, will be gone, okay? Because they are made, they will not be available forever. They also will be gone. So after 7 days, 10 days, we are become better. We are, become healthy again. We cure from the disease. Now, when the second chickenpox virus infection coming, after several months, for example, then we will not perform this process again. Because what? Because we have this memory cell. The memory cell will recognize it, that chickenpox, and then they will destroy it right away. So we don't have the disease anymore. This is what we call the immunity. So we become immune against chickenpox after we have the infection. But if the one that coming, the next one is coming is polio virus, for example, then we have to perform this process again. Okay? So we have to perform this process again, special for polio viruses, and etc. For other viruses, then they will be have the similar process like this one. So this is how our body exactly protect us against the specific infection. After the infection, we fight them and we become cured from the disease, and at the end we become immune to that specific disease. Okay, so that's how the T cell and B cell works in our system. Now, right now we have what we call the vaccination. So vaccination is almost similar with the infections of the disease, but this vaccination is considered as a weak pathogens. Okay, it will cause our system to produce memory cell without having the illness. Okay, so this is what we call the artificial active immunity. So after we have this vaccination, then we become immune to that disease. Okay, like for example, chicken pox vaccination. So the kids that have chickenpox vaccination will not have the chickenpox disease, but their immune system able to produce their memory cell that will protect them against the real chickenpox, okay, viruses. Okay, so the vaccination is actually the weak viruses that is injected into our body. in order to produce the memory cell. Now, vaccines can be prepared, maybe the whole cell that being killed or become weaker. Live cell that is weaker, eh, maybe part of the antigenic molecules that coming from the cell, bacterial cell or viruses, or maybe using the genetically engineered microbe to produce the vaccine, or maybe part of the toxin, the exotoxin that produced by bacteria, converted into toxoid. These are the type of vaccine that is usually used for vaccination. So this is the picture. So the whole cell over here. So the whole virus is actually injected, but that virus that injected is weaker. It's a weaker virus, or maybe just part of of the pathogen. Like for example, maybe just part of the spike of the viruses, okay, part of the cilia or whatever part of the cell wall, for example, of the bacteria injected into the cell. So of course, because this is only part, like just a spike injected into the cell, then it will not cause disease, because the one that causing disease is like this one over here, the DNA. So if only injected the spike, it might increase our production of memory cell without giving us the disease. Or maybe using the recombinations, using bacteria to produce special vaccine. So this how the vaccines is actually being made. This some requirement for effective vaccine. It has to be low levels with low adverse side effects or toxicity. Should protect against the exposure of natural pathogens. Should stimulate the B cell and T cell responses, which is creating the memory cells. Okay? And should have a long-term, long-lasting effects, and should not require higher dose or maybe booster. That's the better vaccine. Should be in infancy. It's not expensive and can be easy to administer. Maybe oral vaccine or injected vaccine. Administration mostly injections. That's the most common type of vaccination. Some vaccine also use the oral administration and maybe nasal administration. Now now we're gonna talk about the immunity. So immunity is the uh, condition when someone is actually immune against specific disease. Okay, there are several type classification, which is active immunity. Okay, this is when the person actually have or challenged by the antigens directly. It will stimulate the memory cell and creating the immunity. And usually, if it is active, it's become long-lasting. Passive immunity, this is actually when the person receives the antibodies. There is no production of memory cell, so just receiving the antibodies. Therefore, it becomes short term because antibody is not forever, right? Maybe 3 months, 6 months. Natural, if it is the challenge is actually part of the natural disease, like for chicken pox disease, that's become natural. Artificial, if it is given through maybe injection, like vaccine, for example, that's become artificial. Now, if we combine them together, then there are several types of immunity. The first one is called the natural active immunity. And this is natural, is mean from the natural cause, for example, chickenpox disease. So after chickenpox disease, and then what happened? We will produce the memory cell. It's become active immunity. So therefore, natural active immunity will be long-term immunity. So this is after getting the chickenpox disease, we produce the memory cell, and then we become immune for a long time, okay, against the chickenpox, for example. The natural passive immunity, remember passive mean antibody. So this is actually someone that receive antibodies naturally, from natural cause. For example, children, baby that receive antibody from his mother during the embryonic stage through the placenta, or during the breastfeeding. Because breast milk is actually have antibodies on that, and this becomes short-term immunity because antibody is not forever. So it's short-term, but it is given through the natural cause. Artificial active immunity. So artificial means injection. So it's going to be acquired through injection or inoculation, like vaccination, for example, or flu shot. This is artificial. It's become active because the one that is injected is actually part of the microorganism. So our body will create the memory cell. It's become long-term immunity. Okay, so chickenpox immunization or vaccination will create the long-term immunity, just the same like as coming from natural active immunity. Here, artificial passive immunity, this is through injection of antibodies. Okay, injection of antibody. For example, if nurse taking care of hepatitis B patient, usually he will receive the injection of hepatitis B immunoglobulin to protect him against the hepatitis B disease. And again, it's going to be short term because antibodies are short term, right? About maybe 3 to 6 months. Okay, this is the picture of that type of immunity. If it is natural, eh, maybe active, like having the disease, or passive, which is the kids receive the antibodies from their mother. If it is active, it's going to be long term. If it is passive, it's going to be short term. The artificial is injection. This one is vaccination, which is injection of the disease, weaker disease. Then it's become active, it's going to be long term. Passive is just like this kid receive antibodies, injection of antibodies, it's become passive. And this passive is going to be short. So short-term immunity for passive. One more is about herd immunity. So when many people get vaccination, then the whole community is become protected. Even the one that do not get vaccination will also get protected because most people is actually immune against the disease. So this is what we call the herd immunity. So the herd immunity, results from uh, mostly vaccination. When most people get vaccinated, then the whole community will be protected against that disease. Okay, that's what we call herd immunity. I think that's all for chapter 14 and 15. We'll see you again with the next chapter, which is chapter 16, disorders or abnormality in the immune system. Goodbye.